We're delighted to welcome Dr. Chaturanga and Bandara and Professor Liane G. Benning from the GFZ Helmholtz Center for Geosciences in Potsdam, Germany. So Dr. Bandara is a scientist specializing in advanced imaging approaches, and Professor Benning is an expert in interface geochemistry and environmental mineral microbe interactions. And together they are developing innovative cryogenic imaging workflows to explore life within ice, combining field sampling and cutting-edge microscopy to reveal how microbes and minerals interact in glacial environments. And today, we're very happy that they will share their exciting work on imaging light-absorbing particles and microbial communities preserved in Greenland ice. So please, Liane.
So thank you very much, Celine, for the introduction. And basically, Chatu and I will try to put you in the next 20 minutes to take you away from your lunch table and bring you over to Greenland at the beginning to show you why we're doing this, and then to show you what we're trying to do. So basically, if you think about snow and ice, normally you think about a white surface if you have skiing or if you know about a nice winter before it becomes gray. What you see here is a scientist with all looking at the departing helicopter, which will not come back for 3 weeks. And that's where we actually try to look. Now, this is the way people think snow and ice looks like. But when the snow melts, sometimes it actually can turn black. And this blackness comes from the fact that you have obviously mineral dust, you have black carbon soot, which we produce by anthropogenic processes or wildfires. But also we have a lot of microbial systems that actually participate in this blackness, as I show you in a moment. Now, so we want to understand what makes this darkness, because the Arctic, specifically Greenland, melts 4 times faster than the average global. The average temperature rises 4 times faster, and ultimately this will be a big problem for sea level rise. But here we're talking about microscopy. So if we want to image what happens on the ice, normally, as you most guys know, when you eat ice cream, it actually melts very fast. So that means you take a piece of snow and ice, and by the time you actually look at it, it has changed, it doesn't melt. It means you lost the coherence. So what you see on this slide is this is our, for example, our field camp, and this whole darkening area here, if you look at it at 2 centimeter scale, you see a lot of black little dots here that actually are part of the minerals, microbes, and black carbon darkening. So if you take a little core, you see the surface ice crystals here are covered by black stuff, and we wanted to figure out how can we image that without changing it. The reason is because we have shown over the last couple of years that specifically biology, and these are pigmented algae, they are either colored green because they have chlorophyll, or all the way to red or deep purple. And this is part of an ERC synergy grant, where which is called Deep Purple, where these algae are smarter than us. We have to go and buy sunscreen. They produce their own sunscreen, but their sunscreen is dark pigment, which colors the ice even darker. Now, the picture that you have here is of a melted sample, where you still see minerals and microbes and lots of other things, but this has been melted, so we lost coherence. So our question was how to do that without changing the sample. So we wanted to investigate the relationships by using cryomicroscopy. But there is no workflow to actually do this analysis, to actually take samples from the ice and bring it back in the lab and image it without changing it. Most cryomicroscopy processes that you hear about and that you see are actually where people freeze something in the lab and actually image it because they want to stop a process. But we had to stop the process from melting in the first place. So what we did is we actually went to Greenland, we collected samples directly on the ice, but before, this is now the ice surface, before we collected them, we actually used a handheld microscope to actually image whether we have algae and minerals on them. Then we collect them in specially designed system that actually kept everything cold. We kept it in a freezer. The ice normally, when we collect this, somewhere between about 5 degrees, and at depth it goes sometimes -2, -10, -20, but it's never -20. But we only had a -20 freezer. Ice didn't recrystallize. In the field lab, we kept it at -20. We managed to figure out how to ship it back home frozen at -20. We then imaged it in a cold room at -20 again with our stereo microscope. And then we designed a system to bring the sample that was frozen still into the cryomicroscope to image it not at liquid nitrogen temperatures, but actually at 20 degrees. That means we try to preserve the whole chain. And I'll only show you a couple of examples, but in order to do this, we first have to design a new system that allows us to do that. This is a sample holder where we could put a 3 millimeter little sample as preserved, both orientationally, we preserve that into the microscope, close the shuttle, go from the cold room, which was at -5 degrees, bring it to the SEM, which was already preset, and then image it. So what you see here is, this is my last slide as well, is this is the image of the handheld microscope at 200 microns. These little filaments here are little algae, glacial ice algae that are covering the surface. Then this picture is from the light microscope in the lab, in the cold room at -20 or -10 actually. At 100 microns, you see the individual filaments here, and this is the ice crystal. And my last two pictures is now the cryo-SEM images where the same samples was actually imaged with, this is the ice matrix, the ice crystals, and these are the filaments of our algae. Obviously it's grayscale because it's SEM, and the minerals interacting, respectively a glacial ice algae, which is different segments, and its EPS, which is the exopolymeric sugars, on a background of glacial ice. So we have done this, we have developed this workflow, and we actually figured out how to do this with samples directly from Greenland to the floor. This is not something which took 5 minutes. This took about 3 years to develop. But what we don't know is actually, how do they look inside? What happens with them inside? How can we reproduce this and actually do it better in a different way that we can actually also look at, for example, ultrastructure to understand a different kind of process. And that's why I'm passing it over to Chatu, because he will more or less take you from that part to all the way to how to image algae in a different way. Chatu.
So continuing from this work. So as Liane said, like we have huge fields of collection of our samples, but then at the end, we only want to see a single cell. So you see the scale from acres to from 10 micrometers and then to the features of 5 nanometers. So either we have like full samples collecting from there. Also, we have some samples growing in the lab that we want to see how they interact with the minerals and everything in microscale. And this is where the electron microscopy workflows we have, and we are going to discuss. So today I'm going to show you the basic electron microscopy steps that we can use. So the one pathway is the traditional way of fixing, post-fixing, dehydration, and then image them. Or if you want to see the ultrastructure, we go for resin embedding and then do ultramicrotomy and TEM or SEM. So the next way of doing it is like keep them in all cold temperatures so that we preserve the water, and then they are in a vitreous status, so they're like completely frozen, and then we don't lose the water content around the cell, so that we want to see minerals, bacteria, or the fungi, and then we can see the entire things as in the nature. So I'll be giving a couple of examples, so how we're going to achieve this, and then what we have achieved so far. So when it comes to sample preparation, so there are a couple of things we have to see, because if you want to see the morphology, or do you want to see the elements of the minerals, or do you want to see the ultrastructure, it would be nice to see everything at once, but unfortunately we can't do that. So we have to make a compromise, and then we have to find a suitable way that we can make a coherent like picture of what is happening there. So, and here's an example what we see. So here we want to see the calcium minerals inside one of the algae. So on the left-hand side, when we go with the room temperature preparation, you see we can preserve the ultrastructure of the algal cells, but we lose the mineral information of the cell, which is telling us this. If we go at the ultra temperature, like room temperature preparation, it's not working for us. On the right-hand side, you can see here we can preserve the calcium carbonate, but we cannot preserve the ultrastructure of the cell. So now we have to find the balance to see what we want to see at one time, and then how we're going to learn from that to explain the biological processes. So we also wanted to know why these things are being damaged during the preservation, and we realized at the moment we fix the cells for the electron microscopy, everything in the amorphous calcium carbonates are being dissolved. So then there is no longer that we can use the samples. And this is one way we decide, okay, with this sample, we need to go with the cryopreservation techniques. So when it comes to cryopreservation, we have two ways of going: high pressure freezing or vitrification with plunge freezing. So these are like standard techniques, but then I will talk about the basic results we have achieved so far. So what we do is we high pressure freeze one of our samples, and then we make thin sections in the ultra microscope. So here you can see in the chamber, and then you see the diamond knife, and then you see the sample, and then we shred the surface, and then we want to cut like very thin sections of the sample so that we can see the ultrastructure of the algal cells. So here you see the cross section of it, and here you see the copper tube we have, and inside we have the cells in water, which is already vitrified. So then the vitrification, which is a solid, now it holds like a matrix, so the cells will not move, and then we can cut it on the surface. And then what we do is, like, we in the cryo ultramicrotome, we make thin sections about like 50 nanometers thickness, and then we make a full ribbon, and then we collect them onto a TEM grid, and which will allow us to do the imaging. And this is, here we show a kind of a relative workflow, and then this TEM grid can go to TEM and then do SEM or TEM, either we need. So what else we can do, because the cryo ultramicrotome is kind of like a really fragile technique, and then you need to be very skilled, and then, but the question is, like, can we do like a bit more room temperature work? So then in that sense, we try to do it with bring the samples preserved in cryo, but we want to bring the processing to the room temperature. And this is what we did with the high pressure frozen samples with low temperature embedding. And you can see, so we have the high pressure frozen sample, we don't see any cells, everything is embedded, and then now everything is into the resin blocks. Now this we can handle in the room temperature, which is much, much convenient for me, because you don't have to keep everything in cool, and this is really flexible with our workflows. So the same principle applies, but then these plastic resins, we cut them into thin sections, and then we collect onto SCM or silicon grids or TEM grids as usual, and then we go to image. And then we could see the minerals we want to see inside the cells are being preserved, and at the same time we can see the ultrastructure. So here you can see the calcium particles within the mineral. So if you compare the previous version that we use only room temperature, these are gone. But if we use only the high pressure freezing, then we cannot do all sort of things we do. But by combining cryo techniques, bringing into the room temperature preparation, we could see the minerals, and then this is much easier for us to work on them. And then we also wanted to see the elemental mapping, and then we can confirm the cells are actually having calcium minerals even after resin embedding. So this is a low temperature resin embedding, therefore we can see the minerals and the elements of the cells and all the particles within the cells. And we also did that with the cells and minerals, but then what we did was, like, we did the fully resin embedding in room temperature, and this is with the minerals, because now you imagine you have the cells, which is softer, you have like minerals, which is harder. Now we need to cross section and image them. On the left hand side, you can see a thin section of the same process we did, and then on the middle you can see the algal cells and the flower like the outer structure, and on the right hand side you can see the algal cells associated with many other cells. So there are so many things, because these are coming from the environment, so then you don't have the controlled environment, but in the environment there are so many things that we do not know. There are like so many particles, and this allows us to do the further investigation of the analysis. So, and this is the second way of doing it, is like, rather than, we also wanted to see, okay, so now we did like cryo and then bring it to the room temperature and see, and then we also want to see that, can we do this in full cryo? This is a bit tedious, but we want to see whether we can achieve these goals. Therefore, we want to do the cryo-FIB SEM and then image the 3D structure of the individual cells. So what we did was, we froze the sample without going for free substitution. We find the cell position and we cross-section a single area, and then we try to create a 3D volume of the single cell. So here I'm showing you is the small 3D recreation of the cell that is going through the entire process. I wanted to show the video so that you can see this is the distribution of the calcium particles within a single cell. So we want to see the distribution, how they are distributed within the cell, because for TEM, we can only see a small part of the cell. But in this approach, we can see entire 3D volume, and then we can see where these particles are located within the cells. And this is one way of trying to see where these particles are within the cells so that we can do further studies. Now, at the moment, we are doing like to establish the workflows, which is suitable for multiple approaches of our working. And the third one we wanted to do is with the plunge frozen samples. And here we see we vitrify the samples, and then we go to the SEM, and then we wanted to see where these particles are and what are the elements we can see within the cell. So here you can see the entire vitrified sample with these cross sections in the TEM grid. And then individual grid has multiple cells, and we want to target a single cell and then see what we can see from there. And on the left-hand side, we have picked one single algal cell, which is about 10 microns here. And then we try to do milling the cell from top to bottom so that we can see how these are being organized. And then we started to see these kind of small round particles coming up. And they're really like rigorous. Even with the ion beam milling, we could not really see they are being disappeared. So they are like really tough. We are currently investigating what these things are. Even after full milling, you can see the bottom of the grid is already consumed, but these particles are still being remaining. And this is one of the questions we want to understand what is happening. And then we are currently developing on this direction in our work. So this has been quite a bit of work involved. So I would like to acknowledge all the team members of our geointerface chemistry, because there's a lot of microbiology, microscopy and field samplings are involved. Also, we work in collaboration with Emmeline and then you from MPI Golm, and of course the funding agency to make this work possible. And of course, we always be happy to be in the Excite network and collaborate with the people, and also for this opportunity today to talk to the Excite community. Thank you.
Thank you very much, Chatu and Liane. Very nice images, and I'm really curious about those little particles there. And so my first question would be, and please, if you have any questions, put them in the chat or raise your hands, and then I can give you the floor. But I'm very curious about those particles, and you already mentioned that you are investigating them, of course. So is this really like a surprise finding to find these kind of particles, and then also the localization in the cells? Is that really something new because maybe you have found a really nice workflow that you have preserved, that you could preserve kind of like how it was in real life?
Yeah, so especially the particles which I showed in the last bit is quite exciting, because these things we could not see when we do it with the room temperature preparation. So that gives us a kind of impression that we might have changed the sample, and then when it comes to cryopreservation, it's exactly the same native state. I mean, very native. So this gives us more excitement and to see what are they. So this, we are currently curious, and then it only shows this is just carbon. That's also again curious, like what are they?
I think that was the most surprising thing, because normally when we looked at it, it's just all the particles that are there are just made of carbon. So you would assume that when you do the milling, the carbon would go the same at different parts of the cell, but it doesn't. So this must be a different conformation of the carbon, or a different kind of binding environment that we have to explore in a different way. So we are thinking of doing sort of soft X-ray microscopy to actually map the different moieties of carbon in these cells after they have been milled, and before and after, but it's still some work to go.
Yes, of course, because now we come to the fun part, right? Yeah, really
well,
great.
It's as usual, because you're looking at for one answer and then you find 10 more questions.
Yes, indeed. So I see that is somebody who would like to ask a question. If not, somebody is typing, which is very interesting as well. So let's go for another question. So for researchers interested in applying this approach, what kind of infrastructure expertise is essential to get started with these kind of exciting techniques?
Well, if you want to start with this approach, my suggestion is go and work with people who know what they're doing. Unless you have 5 million euros lying around and you can buy a cryo microscope, you can buy a bunch of people who know what they're doing to actually do it for you. But actually, when you want to start with something like that, go and work with people who do something like that. I'm not saying come all to us because everybody has different questions, but you also have to be very prepared to fail a lot before you succeed. Because it's not something, I mean, cryomicroscopy has been around for a very long time, but it's very often used, for example, for figuring out how proteins are, sort of protein crystallography, or more and more these days, or for example, for virus imaging, because if you have 10,000 viral particles that you image at very high resolution, you can reconstruct a very high resolution image of a single virus particles. But the assumption is all your particles are the same. Nature mostly doesn't do us that favor. Nature does complex materials. So that means the kind of materials that we image, we can't just take 10,000 images of the same thing and reconstruct. We have to have one cell that we look at in a different way to reconstruct. So it's always depending on the question. But I think it's an exciting frontier because more and more we realize that every sample, as an earth scientist that goes in the field, that we take a sample from somewhere, we do something with it, and then we think microscopy is the magic solution. It is not, because you change it, you put it in high vacuum, you dry it, you grind it, you preserve it. So always think about what do you do your sample and what does the image or the analysis actually tell you about how it was in the field or in your experiment. But there are ways forward because more and more of these workflows are being developed.
Fantastic. Yeah. Yeah, it's always a challenge indeed with microscopy. So I've got a question.
It's also
here.
an opportunity.
Yes, well, definitely. It goes to show. So I've got a question here in the chat. So is it at all possible to subsample these different kinds of carbon chemistry and study their organic geochemistry, so with isotopes or ramped pyrolysis?
So by pyrolysis, not necessarily, because pyrolysis, as far as I know, is mostly a bulk technique. So you can't do at a nanoscale, but we use scanning electron transmission electron microscopy at the synchrotron, where we can actually map at a 30 nanometer resolution the carbon moieties, for example, in one of these samples. You can also do that by other methods where you, for example, potentially use nanosims to actually look at some of the things, but that's equally destructive. So you have to balance what you destroy. It's also, for example, for nanosims, a sample that is very, very thin may not have enough counts to be able to give you a signal. So every method has its good, its bad, and its ugly.
Yep, so we are currently trying to localize them and then get them as TEM lamellas. But then obviously the challenge here is like, we don't see the particle before milling into the cell. So we are doing it blindly. So if you can see them first and then mill it, that's a different story, but you don't see, and then you have to mill it, and it's like a kind of a blind shot. Mm-hmm. So we are trying to extract them and then do it with TEM at the moment. So this is the workflow we are currently based at. Hopefully if it is successful, and that's the only thing that we can at the moment to localize, because they are kind of covered inside the cell.
But I think the question is not necessarily just about our cells, but for example, if you would have a kind of cell and you know a lot from the pyrogenic organic geochemistry analysis, you know about a lot of the bulk microbial organic matter composition, that is really, really helpful. Because if you then have a method where you can spatially resolve the chemistry, you can then take the bulk analysis and combine those two. There is always one single method doesn't give you the answer. You always have to have other methods to help you put your samples in context and your numbers in context. So yes, we do analyze, for example, the particulate organic carbon composition of our cells by digesting them and doing, for example, mass spectrometric analysis to get the composition and the moieties and all the distribution. But most of the time, then we dissolve the whole cell. So what we want to do is single cell spatially resolved in cryo with samples from Greenland. So not a big call, but you know, you have to don't dream big, you don't dream big.
Yeah, yeah, definitely. So I think this question also is a bit related. So do you have any interest or need to connect your nano to microscale information with the ice microstructure? For example, an in situ study looking at relationships between the microbes and crystal microstructure.
Yes. So when we started with this, before Chato arrived, we actually wanted to figure out whether the microbes that we see in the field are actually covering the ice crystals together with the minerals, or are they also sticking in the air? Because these are photosynthetic algae. And that means one of the questions that we always had, how do they disperse across the Greenland ice sheet? So we had one of my colleagues always used to say, these algae are flying. And we didn't know how they are flying because obviously there's a lot of wind. But basically then when we imaged them, we saw that in the frozen state, many of them actually stick up, that they actually fly. Now, some of them actually what they, in addition do, they actually use some nutrients from the ice, from the melting ice. That means phosphorus and nitrogen that is entrapped in the melting ice, and or solubilized minerals to extract phosphorus for the metabolism. Because ice is a really what is called oligotrophic, poor nutrient environment. But these guys don't need a lot. They need very little. They are very frugal. They don't eat a lot, but when they have summer, they have a party and they just bloom. But one of the problems that we have at the beginning is that the first set of samples, we preserve them in liquid nitrogen. But that actually then recrystallized our snow and ice. And that means the reason why our first workflow was just at 20 degrees is because we didn't want to have the ice matrix on which the minerals and the microbes sit recrystallize. But as you well know from your fridge at home, if you have a fridge and you open the door too often, the humidity from the room forms ice crystals inside. So we had to even avoid to change the humidity from Greenland to Potsdam from one room to the other, because you create new crystals that actually crystallize from the humidity. So there are lots of challenges that you have to overcome. But ice recrystallization is a very big problem if you want to study something living on ice. In Chato's cryo work, very often with the samples from the lab, he's using more low temperatures because most of the systems that have been developed for liquid nitrogen or liquid ethane temperatures. You can't do them at minus 20 degrees yet. I'm always saying yet. Eventually you will be, because not many things don't change. If you have an ice crystal, it changes as you drop it in liquid nitrogen temperature at minus 196 because it recrystallizes. If you are interested in the matrix, like the question was before, yes, you have to preserve the matrix as well with the cells, with the minerals, without changing everything, because otherwise you're answering one question, but you don't know how the other one changed.
Oh, exactly. Thank you very much. So here it's thanks and interesting stuff. Thank you for the clear answer and great presentation. Very cool science. That is a joke that that I like. Very cool science.
Thank you very much.
If you would say, what are the next step for the research team? You were talking about TEM already. So what would be also kind of like a fan out for this workflow?
So as I said, one would be the TEM and then going for the SIMS to analyze what elements are there. And also we are quite interested on soft X-ray tomography in cryo samples, so that you can see the 3D distribution within a cell. And this is something we are really targeting at the moment. So we are having discussions with a couple of groups who can do that, and hopefully we will get there very soon.
So it's a combination of looking at a cell in all kinds of dimensions, but also analyzing all the elements, including carbon moieties. But also, for example, when we have the cells which have calcium carbonate, to look not just at what the calcium, whether it's calcium there, but what's the crystallinity. That means we would also like to mix it maybe with Raman or with diffraction or something like that, because it's always the multimodal systems that actually can tell you more. Is there a limit to what we want to do? No. Limits are not what scientists do. You need to dream big, but you need to also dare to spend time to fail. And I'm sorry, Chato, failing is a big part of his job until we get it right. But that's also the fun part of it.
Fantastic. Well, thank you very much. Thank you all for joining this webinar. Thank you for this wonderful talk. And I would almost come back and do science again after this talk, but I won't.
We will.
I will just work for the Excite Network for now. Thank you very much also for your questions, and have a great day. Thank you.
Thank you, everybody.
Bye-bye.
Bye.